The sliding filament theory. Hi, my name is Angelika and today we'll be talking about the sliding filament theory. This theory explains how the muscles contract in our body. In our body, our muscles contain bundles of muscle fibers. Those muscle fibers have muscle cells, which contain a nucleus, or plasma protein, and a smaller unit called a myofibril. Within the myofibrils are the sarcomeres. The sarcomeres are what we are going to be discussing today. This is where our muscle contractions take place. The two proteins responsible for our muscle contractions are actin and myosin. When a muscle is resting, these two proteins do not touch. When muscles receive a message from nerves, the two proteins, myosin and actin, touch. The head of the myosin moves towards the sarcomere, thus creating a contraction. Along with the actin and myosin, ATP is also needed to create energy. At rest, proteins, troponin, and tropomyosin are intertwined and wrapped around the actin, along with ADP. At rest, there is a buildup of calcium ions. They will be used in the contraction. The cell gets these calcium ions from the sarcoplasmic reticulum found wrapped around the sarcomere. Muscles receive a signal from nerves, and the signal travels down the cell's membrane and folds, referred to as T-tubules. Once the signal reaches the sarcoplasmic reticulum, the calcium ions open and calcium ions flow outward. The calcium ions bind with troponin, causing the tropomyosis to leave the actin, thus exposing the activation site. With the binding sites open, the myosin heads attach to the actin. ATP is used, and in the process, it gets changed to ADP. The myosin heads bind toward the center of the sarcomere, a shortening of the sarcomere, aka a contraction. When there is no more stimulus, everything goes back to its resting position, aka muscle relaxation. Thank you for watching my video.